Mesothelioma is an aggressive type of cancer that develops from the mesothelium, the protective lining that surrounds many of the internal organs of the body. The majority of mesothelioma cases are caused by exposure to asbestos. Unfortunately, mesothelioma remains difficult to treat and the prognosis for patients is generally poor. However, researchers have been investigating novel drug targets that may lead to more effective treatment options. Here are some of the most promising drug targets currently being studied. Epidermal growth factor receptor, EGFR. EGFR is a receptor tyrosine kinase that is involved in cell proliferation, survival, angiogenesis, and metastasis. It is overexpressed in a significant percentage of mesothelioma tumors. EGFR inhibitors such as erlotinib, gefitinib and afatinib have shown anti-tumor activity in mesothelioma in preclinical models and some early-phase clinical trials. Larger clinical trials are still needed, but targeting EGFR remains a promising approach. Vascular endothelial growth factor, VEGF. VEGF promotes angiogenesis, which is important for tumor growth and metastasis. VEGF is overexpressed in mesothelioma and high levels correlate with poorer prognosis. Bevacizumab is a humanized monoclonal antibody against VEGF that has shown anti-tumor effects in mesothelioma when combined with chemotherapy in phase 2 trials. Larger confirmatory trials are still ongoing. Other anti-VEGF agents such as aflibercept are also being studied. Inhibiting VEGF signaling remains an attractive strategy. Histone decyclases, HDACs. HDACs are enzymes that remove acetyl groups from histone and non-histone proteins, resulting in transcriptional repression. Overexpression of several HDACs has been observed in mesothelioma. Pan-HDAC inhibitors like vorinostat have exhibited anti-tumor activity in preclinical models and phase 2 studies of mesothelioma. More specific HDAC inhibitors targeting individual isoforms are also being investigated. Given their novel mechanism of action, HDAC inhibitors represent a promising class of epigenetic therapies. Heat shock protein 90, HSP90. HSP90 is a molecular chaperone that assists in the folding and stability of various client proteins involved in cell proliferation and survival. It is overexpressed in mesothelioma tumors and high levels correlate with worse prognosis. HSP90 inhibition using drugs such as OE922, gantespib and luminspib has shown significant anti-tumor effects in preclinical mesothelioma models by reducing the stability of crucial client proteins like EGFR, AKT and RAF. Early clinical studies are also showing signs of activity in mesothelioma patients. Insulin-like growth factor 1 receptor, IGF-1R. The IGF signaling axis regulates important cancer-related processes such as cell growth, apoptosis and metastasis. IGF-1R is frequently overexpressed in mesothelioma tumors and high levels predict poor prognosis. Preclinical studies have shown that IGF-1R inhibitors reduce mesothelioma tumor growth and augment the effects of chemotherapy. Figitumumab, a fully human monoclonal IGF-1R antibody, demonstrated single-agent activity in a phase 2 trial and is now being combined with other agents in phase I/II trials. Mesenchymal epithelial transition factor, c-Met, aberrant c-Met signaling, mainly via autocrine or paracrine HGF loops, contributes significantly to mesothelioma pathogenesis, growth and dissemination. c-Met overexpression is frequently observed and associated with worse outcomes. Preclinical experiments demonstrate that c-Met inhibitors effectively reduce mesothelioma cell proliferation, survival, invasion and tumor growth. Early clinical trials of c-Met inhibitors like cabozantinib and savolitinib alone or in combination are under evaluation for safety and efficacy. Proto-oncogene serine, threonine protein kinase, PIM. The PIM family of serine, threonine kinases regulate important cellular processes including cell cycle progression, apoptosis, and cell metabolism. High PIM expression levels correlate with poor prognosis in mesothelioma patients. Preclinical models show that genetic pharmacological inhibition of pan-PIM or individual PIM isoforms decrease mesothelioma cell viability, proliferation, tumor growth and augment chemotherapy response. PIM inhibitors are currently in early phase clinical testing. While challenges remain, targeted therapies inhibiting pathways such as EGFR, VEGF, HDACs, HSP90, IGF-1R, c-MET and PIM represent some of the most promising drug targets currently being studied in order to develop more effective treatments for this deadly cancer. Ongoing and future clinical trials will help further evaluate their therapeutic potential, both as single agents and in combination with existing chemotherapy regimens.